All right. Thank you for that introduction, and I'd like to thank the JGI for inviting me to share my work with you. And also thank you all for waking up early this morning to be here. As was mentioned, this work started when I was a postdoc in Michelle O'Malley's lab, and through that work, we've been partners with the JGI, both as a postdoc and now as an independent PI in my lab for the last 6 years or so. And so this talk will be a reflection of some of the advances that have been powered by the JGI, as well as how that's fueling the hypotheses that we're testing and working on right now. Now, before I get too far into the science, I just wanted to bring your attention to this awesome symposium that's happening next month. At Purdue University, we have a very growing and very active community of experts in microbiomes, whether we're talking about animals, plants, soil. And so we're hosting this symposium, and we invite you all to join us. Now, going to the talk at hand. So as was mentioned in my introduction, I was trained classically as an engineer. And so the reason why we're fundamentally interested in biology and these types of organisms is we're very interested in trying to understand what are the more sustainable, more cost-effective ways that we can generate things such as biofuels, medicines, commodity chemicals, and so on and so forth. Now, historically, these things have been driven by petroleum sources, just because we were very good at organic chemistry. These things were abundant and cheap. However, as those resources dwindle, we still need to find a cheap and an accessible source of carbon for these materials. Fortunately for us, biology is very good at capturing atmospheric carbon in the form of plant biomass. And through a number of processing techniques such as high temperatures, so a lot of energy input, or with solvents and acids and bases, we can depolymerize these things into simple sugars that we can then ferment in engineered strains to make a wide array of compounds. However, this depolymerization step of biomass into sugar is actually a very potent bottleneck. It's very expensive. It's very inefficient, and the platform organisms that we use to ferment aren't very tolerant. They're not very happy with the quality of the sugars that we have, and so. In my lab, we're trying to identify better solutions. Are there ways that we can streamline or consolidate both of these processes, the deconstruction and the fermentation, to have a simpler and more cost-effective process? Are there organisms that are better suited for these types of jobs? And if so, how can we actually try to engineer them? Like, what are the tools? What are the basic skill sets that we need to manipulate them? These problems are more striking when you think about the fact that biomass is not homogeneous. Biomass, when we think about it, it varies greatly as we go across the United States and across the world, and that's just a function of climate and what will grow where. And so, for example, in the Midwest, at Indiana, where Purdue is, you might find a lot of grasses, so you might find corn stover, switchgrass. Here on the West Coast, you might find more trees, woody things such as poplar, or on the East Coast, you might find things such as eucalyptus. And all of these things have a wider range of sugar compositions, so the chemistry for how these things are polymerized together, and therefore the strategies by how we break them down will differ dramatically. And that compounds this problem of how do we actually get access to those sugars. Now, fortunately for us, again, biology seems to have the solution. If we look at large herbivores, so if we look at cattle, sheep, goats, elephants, giraffes, you name it, these things naturally forage and exist on these things in the wild. I mean, if you see a goat in the wild, they'll be eating grasses, they'll be on the sides of mountains eating shrubs. They're like the garbage disposals of the animal kingdom. And the reason why these organisms are able to eat such a wide array of substrates is because of the concerted actions of their microbiome. And in particular, there's these anaerobic fungi, so this little alien moonlander thing here that is actually burrowing into the substrate. These seem to be central to the community for diets that are rich in fibers. So when you feed these animals very fibrous substrates, such as what you would expect with woody biomass, these things proliferate. And they proliferate because they grow invasively into these substrates, so they create this extensive rhizomycelial network that physically penetrates and tears apart the material. They secrete a wide array of enzymes, and they also secrete metabolites and actually form physical linkages with the archaea, the bacteria, and other components of the microbiome to degrade these things very efficiently. In my lab, we've gotten very good at isolating these things, and we've characterized them on a variety of substrates. And while there's a lot going on here, I just wanted to highlight one thing. So if we look at growth in a variety of substrates, and here is pressure, which we use to track growth. This first bar here in gray is glucose, right? So glucose, pretty much the default carbon source for most organisms. That's probably the most efficiently that the organisms will use it. And however, we see that if we go into crystalline cellulose, if we look at C3 grasses such as alfalfa, C4 grasses such as corn stover, sorghum, and even woody biomass such as poplar, that growth seems to be comparable to what we see on glucose. So what that tells us is that the hydrolysis is not the rate-limiting step in these reactions. The ability of these organisms to degrade these things is comparable, if not faster, than their ability to uptake those sugars and metabolize them. And so it would seem that these organisms are hyper-efficient at this. So how does that work? Well, when we started, there wasn't really a lot known about this. So just to kind of give you some more background about what anaerobic fungi are, here is a fungal tree of life. The later diverging species up here are essentially mushrooms, ascomycetes, things such as Saccharomyces, aspergillus are kind of in the middle here. And here we have Neocallimastigomycota, which are all anaerobic fungi. So these organisms are unusual in the sense that they're the earliest diverging free-living form of fungi. The other earlier diverging strains tend to be parasites or endosymbionts. These until their discovery, people thought that fungi could only be aerobic. And so there's a lot of unusual things about it. However, with recent discoveries, there's been a renewed interest in this class of organisms, and we've grown from roughly 6 genera of fungi in this family when I started to more than 11, and that seems to be growing more and more. However, there's still really no model strain. They're very difficult to cryopreserve. Very few labs in the US are able to cultivate them. And so we have to go back to nature. And so here, being at Purdue, we're in Indiana. There are many farms around us. You can see here my undergrad and one of my graduate students here. They're working with these animals here where they patiently line up to do their part for science. And so we can collect these isolates and we can study them. What many have found over the years are that their genomes are very AT rich. They're more than 80% AT, so less than 20% GC. So there's a lot of homopolymer repeats, which poses some unique challenges that the JGI has helped us overcome. And they're not quite yet genetically tractable, but hopefully by the end of this talk, I'll convince you that there's hope for that. So when I started my postdoc back in 2012, 2013, there really wasn't a wealth of molecular information. I mean, there was an extensive community of animal microbiologists, so those looking at nutrition in the animal gut, and they had done some beautiful work looking at the metabolism and physiology of these organisms. But no one had really been able to probe deeply into these organisms because genetics weren't developed yet. Next generation sequencing technologies were just coming online. And so the only thing we had available to us was this organism called Orpinomyces C1a, which later became reclassified as Picomyces. You can see that we had a genome that was fairly large, so 100 megabases, only one transcriptome, but you can see the quality of the genome was quite poor, right? I mean, at the time, we really only had Illumina sequencing, so we had these really, really short reads, but with these homopolymer repeats, it made it very, very difficult to reassemble and reconstitute this genome. However, through our partnerships with JGIEMSL, we have greatly expanded on this and refined our understanding. So now we have transcriptomes, proteomes, genomes, and metabolomes for a number of species. The introduction of pack biosequencing, among other technologies, has allowed us to take longer reads so we can actually get higher quality assemblies. And so while we still don't have a complete genome, we can get this down to roughly about 200 contigs. So we're advancing towards better resolution, and I think now the challenge here is on the biochemistry, biological side. So how do we generate better, higher quality samples so we can have larger fragments to aid in assembly? And going forward, we have a solution that we think will help with that. So through our partnership, we've characterized more than 4 species. You can see their genomes range in size from about 50 megabases to 200 megabases. And there are many stories to tell here, but for today, I'll share a couple of those vignettes and then show how those are powering the studies and the science that we hope to accomplish in the future with the JGI's help. So when we first started, as I said, we didn't really have a genome, right? We didn't have a genome, and we knew that this would be challenging. And so rather than taking a genome and then using that to develop a transcriptome, we decided to go the other way around and acquire a de novo transcriptome, with the idea being this would allow us to really focus on the business end of biomass degradation. If we grow our fungi under relevant conditions, such as with lignocellulose or without, we would be able to not only generate a catalog of the enzymes that were involved in generate the catalog of enzymes involved in biomass degradation. We would be able to root out introns and other things that would confound our analysis, and more importantly, we'd get a sense of for which ones were actually important. We would only look at the ones that were active and not the things that were silent or cryptic. And so one of the first experiments we did is we had this hypothesis that because biomass degradation is non-trivial, there are a lot of enzymes that are involved, that when we grow our fungus on glucose, there would be catabolite repression that would repress those enzymes. And so just by looking for what things were missing, we would be able to find out what were the relevant enzymes for expression. And so here I'm showing you data where we're looking at the expression of biomass degrading enzymes in blue, as well as their activity in red. And we can see that after growing these organisms and then spiking with glucose to initiate catabolite repression, we see a dramatic drop off in the expression of these genes. And roughly after 3 hours, when most of that glucose is exhausted because we only added a little bit, we begin to see some recovery of the expression. We can look at this in more detail and look at a heat map of genes that are differentially expressed. And here we're looking at things that show at least a twofold difference with a p-value of 0.01 significance. And we can see that there are genes that naturally cluster into these regulons. And more importantly, these regulons are enriched in function. So not only do we see a nice dynamic remodeling of the transcriptome, we can actually begin to ascribe them to function to begin to understand how anaerobic fungi degrade these materials. And so one of the more interesting things that we saw, which in retrospect is probably a little bit obvious, but it was nice that this experiment was reconstituting what one would expect. We can see, for example, that this cluster here, this regulon here, seems to be more responsive than this one here. This cluster is involved in hemicellulose degradation. And so if you think about plant biomass, you have this energy-rich core of cellulose. It is surrounded by hemicellulose and pectin, and then that's wrapped up with lignin, a structural polymer to hold everything together. And so naturally, you would expect the cells to digest it from the outside in. And so therefore, you would expect them to want to express the hemicellulases and the pectin molecules first, pectinases first, and that's exactly what we see. Similarly, it also helped us to improve our annotation. So if we look at this transcriptome, despite the rich data set that we could get, we could only annotate roughly about a third of the genes. So there are many that we have no idea what they did. However, by looking at how they co-regulate with some of these clusters that are enriched in function, we now had hypotheses for what those novel enzymes could do, and we can then do some more traditional biochemistry, molecular biology to figure out what they do. Similarly, we expanded this out to different substrates. So here we're going from cellulobios to more complex, untreated plant substrates. And here we're looking at the expression of different classes of enzymes. And naturally, as you would expect, as substrate complexity increases, the types of enzymes increases as well to deal with that complexity. However, we can also begin to see some more profound insights, such as the fact that as these things go from crude lignocellulose to pure cellulose to even cellulobios, you would expect that the enzymes that are needed here would also be needed here, because this cellulobios, for instance, is essentially a breakdown product that results from any of these substrates. However, what we see is that there are subtle shifts in the types of enzymes which suggest that their preferences are dependent on substrate that perhaps might be useful when we think about how to create more efficient enzyme formulations. Similarly, we see there are core groups of enzymes that don't show any regulation at all, and we suspect that these are scouts or sentinels that the cell continuously secretes so they can solubilize, at least to a small extent, their biomass, any biomass that they can encounter to essentially generate the signals that then will upregulate these responses that we see. And finally, we can also get some insight into the biology of these organisms. So, for example, we see groups of antisense RNA. We see other types of regulatory molecules that we believe help to shape this response. And so, at Purdue, one of the things that we're doing is we're an agricultural school. And so we have access to vast libraries of different types of substrates, and we can actually control that substrate composition with detail. And so one of the things that we're trying to do is to understand what is it about these substrates that allows these anaerobic fungi to respond in this way. And so, for example, let's talk about lignin. Plants have evolved lignin as essentially as a defense against microbial degradation. It's a way that plants have protected themselves, and they've gotten so good at it that they have created different monomers, H, G, and S monomers, which when they combine them in different ways afford different degrees of protection. However, they are very successful in that. And so, for example, if we take look at this type of fungus, if we go from low lignin, so in this case wild type, which is roughly 60% S-lignin, to high lignin, which line 64, which is 94% S-lignin, you can see there is an inverse correlation between S-lignin and degradation. This fungus, like most other fungi, such as trichoderma, are very sensitive to the specific composition of lignin. However, anaerobic fungi, they grow with equal abundance, and if we look at the composition of the sugars that are solubilized, we see that there's no hit in the hydrolytic efficiency. And here, the low S is actually 5% S-lignin, while our high S is roughly 90-odd%. So essentially, picture the extremes of this plot here. So the anaerobic fungi seem to be fairly insensitive to the lignin content, and we can see that this is due, at least in part, to the remodeling of their secretome here. And so one of the things that we're trying to accomplish in our CSP is to understand how anaerobic fungi are able to detect and respond to this. And this is more striking when you consider the fact that lignin chemistry, so the ways that organisms degrade lignin, all rely on oxygen. Anaerobic fungi, they're obligate anaerobes. They cannot survive in oxygen, and as far as we can tell, we don't really see any enzymes that are able to degrade these materials. So it's really curious to me what exactly are they're detecting, and how have they evolved mechanisms to recognize these things when they don't have the chemistry that we know of, at least, to actively degrade these things. Other things that we've discovered with our partnership is that the anaerobic fungi are unique in the fungal kingdom in that they express cellulosomes. So this is a common strategy in bacteria where they secrete a wide array of enzymes. They have these dockerin domains that plug into a scaffoldin that's anchored to the cell. And that essentially puts these enzymes in close proximity and essentially creates a plant biomass deconstruction assembly line. So you're able to get synergistic action by putting them in close proximity. However, through looking at this regulatory data, by looking at our genomes, we were able to find a class of novel, unannotated proteins. And once we did some biochemical analysis, so we actually expressed heterologously fragments of this, we were able to demonstrate for the first time that anaerobic fungi do in fact have cellulosomes. And finally, and I apologize, it's probably too tiny and hard to read, but here we have the fungal tree of life. Again, mushrooms up top, ascomycetes, so Saccharomyces in the middle, anaerobic fungi at the bottom. And here I'm showing you the biomass degrading enzymes here on this axis here, with the colors representing functionality. And we see that our anaerobic fungus actually seem to have the broadest array, the largest diversity of these enzymes, and they don't seem to be particularly biased in functionality, which is consistent with what we've seen on their growth characteristics. And you can compare this to, say, for example, Trichoderma and Aspergillus, which are the current go-to strains for the production of these enzymes. And so it seems that anaerobic fungi, they have many stories to share with us, both in what do all these extra enzymes do? How do they use them to cope with this wide array of substrates? Now, part of the reason for why they have such a large array of these enzymes is due to extensive horizontal gene transfer in the guts of these large herbivores. So we have this very strict anaerobic environment that's very low on energy. You have archaea, bacteria, protists, fungi. They're all trying to compete and try to eke out a living. And what seems to be happening is that these anaerobic fungi, they've been very good at taking up those genes from their competitors and essentially integrating those strategies. And so, for example, if we take this example of the GH10, these are xylonases. We can see if we look at the anaerobic fungi, xylonases, which are indicated in red, you can see in the phylogenetic analysis that there are two clusters, two clades. We have this bottom group here that clusters with other fungi, but we have this top group here that clusters with Clostridia or bacteria. And so these are opportunistic organisms that seem to have evolved to really to steal from their neighbors, essentially, to better compete. So now that we have a better sense for what is there, we have a better sense of what's possible with these anaerobic fungi. How can we develop tools to actually control them for biotechnology? How can we, for example, can we actually identify the exact enzymes that are the more important enzymes for function? So can we do functional genomic screens, for instance? And so this work is in progress, but we've had some very encouraging preliminary results. So when we started, again, there weren't any genetics. And so what we decided to do was to look at the epigenetics of these organisms instead. So just as a brief reminder, anaerobic fungi, they're eukaryotes. So their DNA has structure. It forms chromatin, and depending on how condensed or expanded that chromatin is, that will help determine how genes how the regulation of key genes. And that will control how those genes are expressed. And cells essentially have these proteins such as histone deacetylases, histone methyltransferases that can add covalent groups such as acetyl groups, methyl groups to the histone proteins themselves and to the DNA that the histones are, they can add it to the DNA itself or to the histone proteins that DNA is wound around to essentially mediate that shift between this expanded and condensed form. And there's been precedent to show that, for example, in other species such as Trichoderma or Leptosphaeria, by changing the distribution of those tags, you can change the activity of CAZyme. You can change the distribution of CAZyme expression and their activity. And so we asked the question, what happens when we add epigenetic inhibitors to the system? Can we actually control this in anaerobic fungi? So our hypothesis was that simply that supplementation of these things would globally change the epigenetic state and ultimately CAZyme expression. And so here I'm going to show you Western blots with a positive and negative Saccharomyces control. And here we're tracking two different epigenetic markers, H3K4 trimethylation, which is present in wild-type Saccharomyces, and H3K27 trimethylation, which is absent in wild-type Saccharomyces, as well as have a histone loading control. And when we look at one of our fungal isolates, we can see the default state. We can see what our anaerobic fungi is happy with. However, when we add the SAHA, this histone deacetylase inhibitor, we're able to get this dramatic shift, a global shift in these states. So the H3K4 trimethylation is dramatically upregulated, and H3K27 trimethylation is downregulated. And more importantly, this shift is accompanied by a change in the secretome. I know it's really hard to see here, but what is a little bit more easier to see is the activity. If we look at the xylonase activity of our cultures, and here I have the full secretome and an enriched fraction, we can see that by adding this epigenetic inhibitor, we can almost double expression. And so with our current ficus, we're trying to get a better sense for what are the knobs that we can tune. What is the catalog of these marks that are available to us? What genes are they associated with? And more importantly, what happens when we change them? How does that affect the expression of these genes? Since then, we've also begun to have some successes with genetics as well. So anaerobic fungi, they have this complex life cycle. And so we've been focusing with the zoospore, or the young juvenile stage of this fungus, partly because they have thinner walls, so it's easier to put DNA into them, and more importantly because they're the younger stage. When they mature and reproduce asexually, any genetic changes we make will propagate. And about a year and a half ago, it was also shown that these anaerobic fungi are naturally competent, which we think might be why they have such extensive horizontal gene transfer. And so we replicated these experiments and showed that conclusively that we can introduce labeled double-stranded DNA or single-stranded RNA, so RNAi or standard genetic transformation, and these things localize to the nucleus. We've also been able to show that when we put genes with a putative promoter, and here we're putting this in a bacterial vector, because at this time we don't have a vector for these organisms, and so because this is a bacterial vector, this plasmid will not replicate. But because these cells are naturally competent, what we do is we introduce this plasmid at regular intervals, and what we find is that when we add a selectable marker, so here I'm showing you growth of negative and positive controls for fungal growth, and when you add the antibiotic hygromycin, they're sensitive to that growth, they die. However, when you introduce the high B resistance gene in this plasmid, we're able to rescue that phenotype. And so it seems that we're able to affect transient expression. And we can also see this in multiple genes. So here I have ILOV, which is a fluorescent protein. A protein that's fluorescent under anaerobic conditions. And so using the sequence information, we're able to pull out localization tags. So in this case, we have a nuclear localization sequence. And we show that when we add our ILOV cassette, we're able to increase fluorescence above background. And more importantly, when we add this NLS cassette, we're able to localize it to the nuclei. And so we now have a tool set that we're hoping to build out so we can actually do CRISPR-Cas9 to then make libraries of mutants that will allow us to do more functional genomic screens. So just in conclusion, hopefully I've convinced you that anaerobic fungi are the best things ever. You may disagree, but we can argue over a drink. They're awesome because they have a wide array of CAZymes that have come from a wide array of organisms. These enzymes allow these organisms to adapt to different substrates and overcome inhibitors that many other organisms struggle with. We can control some of this activity using epigenetics, and we've also had some initial successes developing genetics. And going forward, we look forward to our partnership with the JGI and EMSL to help advance these fronts. I think we're running low on time, so I'll skip what I'm going to do in the future, but I just wanted to acknowledge my postdoc group, these people in particular, as well as my own group and these people, and I'd be happy to take any questions.
We have a time for 1 or 2 questions.
Oh, sorry. Yep.
Okay. Yeah, very interesting talk. I have 2 questions for you. One is that clearly this is a very powerful system, and there'd be multiple ways you could use it. One is trying to add a multi-component system where you have fungi and bacteria or other yeast for your conversion of the biomass. And the other one is to make use of the enzymes. So my question is, are you exploring both avenues, or is there one avenue you prefer? And then the other question is, those fungal cellulosomes are really interesting. Those tend to dig holes in the biomass. So do the fungi secrete both free expressed enzymes and cellulosomes? I'm a bit surprised that they have the cellulosomes.
So, okay, so answering those questions in no particular order. So yes, they express both free and cellulosome-associated enzymes. As for strategies for how we hope to develop this going forward, the community is growing, and there are some in the community, like Michelle, my postdoc mentor, who are working more on that community angle that you talked about. So how can we make synthetic consortia that partner these types of organisms? For myself, I'm more interested in how can we use these fungi directly, either as a platform for mass production of these enzymes. So just like trichoderma, over the 40 years we've done mutagenesis, et cetera, to create these hypersecretors. Can we do that in a shorter time with more controlled modifications? Or can we actually directly manipulate them to do consolidated bioprocessing? So forget enzyme production, just go directly to product. Sorry.
Your hypothesis is the enzyme is genes horizontal transfer. So you have based on sequencing information, do you have any idea about those enzyme gene clustered together, or those genes or not?
So you mean like whether they're co-localized, like and so they transfer together?
Right.
So that is not a question that I have tried to answer yet. So I don't. The answer is I don't know. But I think also because of the current quality of the genomes, given the large number of fragments, it's not something I think we can answer with any high degree of confidence. And I think that we'll have to wait for future. Thank you.